So, hello everyone. My name is Kostas Kampourakis from the University of Geneva, and I'm delighted to be able to give this presentation to you. I'm very grateful to the organizers at NORST for giving me this opportunity. And my aim is to present a quite radical argument about removing what I call naive Mendelian genetics from schools. What I mean by naive Mendelian genetics is not that Mendelian genetics itself is naive, but that the version that we usually teach in schools is, and therefore that it would be useful to replace it with something else. So there are two problems about this, which are described in detail in the book that you see in my background, which was published a few months ago. The publisher kindly provided me with a flyer with a discount for those who are interested. And the two problems are what I call anachronistic Mendelism, the way we read history based on what we know today, and social Mendelism, which is about the social uses of Mendelian genetics back in the time of eugenics. At the end, I'm going to present alternatives, what I think we should be teaching in schools. So let me proceed with this, with the first part. I assume that you all know Gregor Mendel and his famous paper published in 1866. That was the paper in which he presented the results of his experiments that took many years to be completed. And what we usually do is to present in schools images like this about how he managed to clarify and first of all, of course, understand how traits are inherited and how different phenotypes, different versions of the same trait based on respective alleles were transmitted from one generation to the other. Indeed, we explained that Mendel could actually understand the presence of alleles that correspond to traits, and we thus assigned to him an understanding that actually did not exist at his time and which we cannot find in his paper. Nevertheless, even in these educational pages of the journal Nature, we see that this is what is presented, that Mendel understood heredity, that Mendel understood that genes are there and that there are alleles control traits. And this is also what we find in popular books like this one, The Gene by Siddhartha Mukherjee, who is a Pulitzer Prize winning author, who says that the gene was discovered by Mendel in 1864 and then quickly forgotten. And even if Mendel had never done anything else, already understanding the particulate nature of inheritance would be enough, because he could understand how traits are transmitted from one generation to another via the transmission of alleles. Mendel, of course, he says, did not give gene its current name, but nevertheless, he understood everything. Another problem with this account is that Mendel is described as a lonely genius ahead of his time. And even though he was a genius, he was not lonely and he was not ahead of his time. He was very much a person of his own time. The problem with these representations is that these stereotypes of the lonely genius are also enhanced by TV shows like that of Dr. Sheldon Cooper in Big Bang Theory, whereas, as you all know, science is a social process that is actually done by communities. I'm going to first look at what exactly Mendel wrote, and I rely on this fabulous translation by Staffan Miller-Wille and Kirsten Hall, who also explain in this book the actual use and the specifics of the German language. So this is very useful to have in order to properly understand what Mendel intended to write. And right from the start, he makes his aim explicitly clear. He wanted to investigate the law according to which differing traits are transmitted from one generation to another. And he insists about his focus on traits, and what he describes as dominating and recessive, what we would call today dominant and recessive, are the traits themselves and not any alleles or underlying factors. He is very clear right from the start that he's interested in traits, and he makes no speculation about how these could be determined, even though he had the understanding realized that there was something within the organism that was responsible. And this becomes most evident when you compare how Mendel presented the results of his work, which you see above, to the usual representation that we find in textbooks today, which is the one below. Exactly because Mendel was thinking about traits, he presents a complicated ratio which has to do with all kinds of traits, including the ones that he described as hybrid. Hybrid trait was something that contained both elements, but only one was present, was evident, was visible because it was the dominant. And his representation here has nothing to do with the alleles that we describe today. In fact, it was William Bateson who reinterpreted the presentation of Mendel's results from this way you see here in this way. And actually, what we teach in schools today is essentially Batesonian genetics. William Bateson's interpretation of Mendel's work, not Mendel's himself. The truth is that around the time that Mendel was working, there was an active community of researchers who were trying to develop a theory of heredity in order to complement Darwin's theory of evolution, starting with Charles Darwin himself and his provisional hypothesis of pangenesis, Herbert Spencer, Francis Galton, and others. And among themselves, these people, it was only the Swiss botanist Carl von Nageli who came in contact with Mendel through a long correspondence. But it seems that Nageli did not find Mendel's results as crucial for any theory of heredity. And there was a good reason for this, because Mendel was actually interested in hybridization and not in developing a broad theory of heredity. So a very important point is to keep in mind that whereas, as we know, science is a social process, not as harsh as you can see here, but still a social process, it is important to refrain from maintaining the stereotype of the lonely genius in our teaching. We should show, as I hope is evident here, that in all cases, and in this one as well, there are communities of scholars, naturalists at the time, interacting with one another, and Mendel is nowhere to be seen in this scene. Mendel was actually working in the hybridist tradition, which can be seen as an experimental program that, of course, led to a focus on individual traits, but it was hybridization that he was interested in, and he was following the work especially of Kallreuter and Gartner, who had done multiple experiments before him, but no one ever ingeniously analyzed his results in statistical terms like Mendel did. So Mendel was really an innovator. Mendel's work was really important, but not for genetics at the time, not for the study of heredity, but for the study of hybridization. And his main influence seems to have been his professor at the University of Vienna, Franz Unger. And a crucial reason, perhaps, for his experiments was, according to Rosalind Wunderlich, that there was a competing view at the time between Unger and Edward Frenzel, another professor, another botanist. These two were not in agreement about what exactly happens in fertilization, whether both parental plants or only one contribute to the new plant that emerged, to the new plants that emerge from fertilization. And actually, Wunderlich goes as far as to suggest that because Mendel was in agreement with Unger's view that both make a contribution, Frenzel did not let him pass his teacher examinations. He was probably his examiner, exactly because he held a different view. And it is interesting to see that in parts of Mendel's results, we do see what we know today as reciprocal crosses. Crosses that show that whether the male or the female plant is one or the other trait, in the end, we get the same results. So according to her, one main goal of Mendel's experiments was to show that both parental plants contribute something to the offspring and that there is a transmission of traits from one generation to another from both of them. Whether she is right or not, we cannot know because it has not been established Fensel was indeed his examiner and whether it was him who did not let Mendel pass his examination for this reason, but it is a quite reasonable and even more interesting scenario. Now, the most important thing is what brought Mendel back to the scene in 1900. Karl Korenz, who was a student of Nageli, was perhaps aware of Mendel's results, and at some point he came across the paper and then subsequently had similar results himself. So he published a paper in a journal after seeing the paper by Hugo de Vries, in which de Vries mentioned Mendel's ratios, but not Mendel himself. Later, Erwin von Tschermach, who was interestingly the grandson of Edward Fensel, followed with similar ratios and similar results, and it was actually him who maintained that Mendel's paper was included in a big book about fundamental important works in the history of botany. But according to Augustin Brannigan, a sociologist who already in 1979 explained this story here, it was Korenz's use of Mendel's name in the title of his paper that brought Mendel back to the scene. And what had happened in the meantime between 1866 that the paper was published in 1900? August Weismann followed the ideas of Francis Galton about a separate germplasm that was independent of the soma, the main body, and also drew on the advancements of cytology to talk about how inheritance works. So all in all, what happened is that 34 years later, Mendel's paper was not simply rediscovered, it was read in a new context. And in that new context, it made a different sense, and the brilliant experimental approach that Mendel used was very useful. Robert Olby has also shown that Mendel's paper was not ignored. It was included and actually cited by several naturalists who nevertheless probably did not find it very relevant for their work outside hybridization. And he went as far as to describe his landmark paper, also published in 1979, Mendel no Mendelian, and asked the question, what is a Mendelian? And he, as you can read in the quotation here, it depends on what we mean Mendelian in order to classify Mendel himself as Mendelian. If we're thinking so as a Mendelian, as someone who is talking about hereditary elements that are transmitted from one generation to the next in pairs, then Mendel was clearly no Mendelian. But if we're talking as someone who studies the transmission of traits and analyzes statistically the results, then Mendel was indeed one. In other words, Mendel had an ingenious experimental approach, but also had a very specific understanding of his work and interpretation of his work, and it was only later that his paper was read in a different way. An important criticism came very early in 1902 by Raphael Weldon, who somehow repeated experiments like those of Mendel, only to find that there was a continuum of variation, as you can see displayed here on the top, from greenish yellow to yellowish green. And as you know, Mendel spent 2 years doing self-fertilization to arrive at pure homozygote traits, which is not bad if you want to establish a model system. But nevertheless, what he actually did by doing that was to eliminate all natural variation, all variation that exists in nature, which Weldon showed that was really there. So the either or characters of Mendel were the results of his method to develop an experimental system, a model system. And of course, this is what we do in science. We develop model systems to study heredity. And it was indeed a system that was later co-opted and was very successful, but they do not describe how actually heredity works. Nevertheless, Weldon's untimely death and Bateson's influence, his 1902 book was a response to Weldon's paper and also included the first widely read translation of Mendel's original paper, made Mendel the basis of the new science that Bateson himself named in 1906 genetics. So we see here that a specific paper and a specific approach was co-opted in a new context, and this was the basis of a new science. But it was mostly thanks to Bateson's reinterpretation of Mendel's paper and to Bateson's persistence and win in the debate with Walden that we have the genetics that we have today, both in science and in teaching. This is all described in detail in this magnificent book by Craig Ruddick, which you might want to read if you want to delve deeper into the details of this story. What is the problem? For me, the problem is that even today, we teach this simplistic Mendelian genetics about dominant and recessive alleles, about capital B that gives brown color and small b that gives blue color, and we tell students that two parents with brown color, eye color can have children with blue, but not vice versa, even though already in 1909, there was such a case reported. There was a case known where two parents with blue eyes had a child with brown eyes. Most importantly, as you all know, the variation that exists in eye color and all other kinds of traits is much more wider than what we describe in these simplistic models. So whereas we teach students how we do science and how we use model systems, we are implicit about this and we pretend to show them how heredity works, which is actually a phenomenon that is much more complicated and a phenomenon that we cannot understand if we do not take development into account. That's the problem, therefore, with anachronistic Mendelism. A misrepresentation, a distortion of the actual history, and the integration of a simplistic model in our teaching, which, unfortunately, as I will show, has had very devastating consequences. This brings us to the second part of the question, which is social Mendelism. The story begins in 1909 with Charles Davenport, who founded the Eugenics Record Office. And even though he knew Galton and Pearson many years before that, he suddenly became very interested in human heredity, and he became a proponent of Mendelian genetics and their application in human heredity. And he insisted in the pattern we just described and that we all know, that brown-eyed children can only emerge from brown-eyed parents, and that those blue-eyed parents who wanted to have brown-eyed children wished for the impossible, maintaining the rule that we usually also present to students. And when he came to that case I just talked to you about of 1909, the one case where a man with brown eyes was born of blue-eyed parents, he put it under the carpet. He described it as a discordant case, probably due to an error, because it did not fit, perhaps, his broader explanation. And things became worse when he met Henry Goddard, who wrote the famous book about the Kallikak family and about how feeble-mindedness, a trait that I think nobody could describe exactly at the time, but nobody even cared what this was about. It was a term to broadly describe people who are not that intelligent and who were perceived as a burden to the society for whatever reason. So Goddard, with his book, established that feeble-mindedness was a recessive Mendelian trait. And starting from the person that he called Deborah Kallikak, who was in the institute in which he directed, he traced her lineage back to several generations, arriving to Martin Kallikak, who had two lines of descent. Before he was married, he mated with a so-called feeble-minded woman. How Goddard knew that she was one remains an open question. And Goddard managed to show that along the way, most of the descendants, or all of the descendants, were feeble-minded. Later, when Marty Kallikak was married to a woman from a noble family, things became eventually very good, and there was no problem in that lineage. And this is actually from these two lines that the name of the family comes. It's a pseudo name. Kalos and kakos in Greek means good and bad. And this is his idea to represent that there were two lines of descent. And this was a book that was taken as scientific evidence for that trait being inherited as a simple Mendelian recessive one. Now, of course, you may be wondering what happened with geneticists. Well, many geneticists were proponents of this view, starting with the person who gave his name to the Punnett squares we use, Reginald Punnett, who wrote already in 1912 in the proceedings of the eugenics meeting in London that it is clear from the available evidence that feeble-mindedness is a simple case of Mendelian inheritance. He had no doubt about this, and that was the case for many geneticists at the time. Very few questioned Goddard's theory, and it was only very later that they started changing their views, but in the beginning, everyone accepted this as science, as mainstream science. And so, whereas nowadays we describe the same thing, we teach the same thing in schools as in Drosophila, with specific alleles determining red or eye color in eyes, we fail to note that the person who founded that science, Thomas Han Morgan, already in 1915, as you can read here, made clear that there is no way that a single allele can determine a single trait. As he explains here, we need more than 25 factors. It's genes that he had in mind, but he didn't yet use the term. More than 25 factors are necessary for the development of eye color in Drosophila. If one of those changed, then it would be possible to have a different phenotype, but that one allele was just a difference maker. In all cases, all 25 genes were necessary. And this is a representation of this and of what we perceive today correctly, genes as being difference maker. They are causes that can bring about differences, but it's never the case that one gene alone is sufficient to determine a phenotype. This was already clear to Morgan in 1915, more than 100 years ago, but we still do not talk about this in schools. And as it was expected, Morgan became annoyed by the application of simplistic Mendelian schemes to human heredity. And as you can see here, already in 1924, he reacted to these simplistic models, and he said that we should not accept acritically every claim that is advanced as furnishing evidence that bodily and mental changes are also transmitted in such simplistic ways. In his 1925 book, he became more explicit and he more clearly criticized those he described as race propagandists who were trying to categorize humans and trying to explain the complex inheritance of human traits with simplistic models. Nevertheless, eugenicists moved forward and they organized seminars, and this is what you see here is Leon Fradley Whitney in a seminar he was giving to people who were working for Davenport and others in collecting data. And as you can see, he uses Mendel's theater and he presents a typical explanation of how two parents with the same trait can have 4 kids, 3 with the same trait and 1 with something else which was recessive. There were eugenic exhibits at the time, and you see here one from 1929 in Kansas, and you can see if you zoom in here how they used Guinea pigs to explain heredity. And for those of you who are familiar with Mendelian genetics, you can see all kinds of combinations here. But this is not the most important. The most important are panels like this that argue that unfit human traits such as feeble-mindedness, epilepsy, criminality, insanity, alcoholism, pauperism, and many others run in families and are inherited in exactly the same way as coloring guinea pigs. So they present all these human traits, some of which are even difficult to describe as traits, as inherited in this simplistic manner and as controlled by recessive alleles. And they go on to say that if all marriages were eugenic, if there was selection among the fit only to propagate, we could have a better society in just a few generations. And they continue, you can improve your education and even change your environment, but what you really are was all settled when your parents were born. This is eugenics. So what I and others describe as Mendelian eugenics was the adoption of Mendelian genetics in its more simplistic form to explain heredity. And the outcome was this. Even though since 1907 there were legislations about sterilization in several states, starting with Indiana, they were not really implemented. What came to be known as negative eugenics stopped the propagation, the reproduction of these feeble-minded people could happen either by isolation in institutes, which was what was mostly preferred, or sterilization. However, in Virginia, at some point, the legislature wanted to test a new law that they had, and they chose Carrie Buck, whose mother, Emma, was already institutionalized. And Carrie, unfortunately, followed her because while being a member of a foster family, she was raped, became pregnant, and ended up being considered as feeble-minded as well, because one of the criteria for feeble-mindedness was promiscuity and other kinds of behavior. It was an all-inclusive thing that was used in the way that people wanted to use it. And this person here, who was one of the major eugenicists in Virginia, Joseph Desornette, explained in his testimony in the trial that simple Mendelian genetics give us the answers. The feeble-minded woman will have 3 children to every 1 child the college graduate will have. They are easily oversexed, and it depends on their looks as to how the boys or men will take advantage of them, and it depends on her opportunities. So the implicit message would be sterilize them to stop this trait being transmitted. And when he was asked what the evidence for this was, he cited the Kallikak family and the Kallikak case, which he considered as crucial evidence for his argument. And he said that this is a report that was generally published throughout most of the books of heredity, and he was right. It was widely cited and used. He also wrote a poem he named Melton's Law, a plea for a better race of men, in which he wondered, why do we spend time to breed our animals as we wish, and we don't do that for our normal populations? And his argument towards the end was that we should implement Mendel's laws and try to breed only the best kind of men, the best kind of humans. Another proponent who testified in the trial was Harry Laughlin, the superintendent of the Eugenics Record Office, a very close collaborator of Davenport, who stated that in the archives of the Eugenics Record Office, there were many hundreds of manuscripts of pedigrees with feeble-minded members. And according to him, they provided all the evidence necessary to establish that indeed feeble-mindedness was a recessive trait, a recessive Mendelian trait. And of course, one way to stop this was sterilizing the feeble-minded people. And so we arrive at the decision made at the Supreme Court, where the case ended up, with Supreme Court Justice Oliver Wendell Holmes claiming at the end that 3 generations of imbeciles are enough. These were Carrie, her mother, and her daughter. And of course, that it was important to try to protect society from these people. And he made an analogy with vaccination. If compulsory vaccination is there to protect us from infectious disease, so compulsory sterilization should also be there to protect us from feeble-minded people. Nevertheless, more and more scientists started to react, and one of those was Thomas Lancelot Hogben in England, who cited the results of a eugenicist in England who was actually blaming the ones in America about oversimplifying things and destroying eugenics, in which it was clearly shown that the results they received from crosses, it was not crosses, it was analysis of pedigrees, were not in agreement with, the one would be expected with the Mendelian model. And so Hogben concluded that it is inappropriate to conclude with such certainty that feeble-mindedness is inherited as a recessive trait when the evidence does not support this. Morgan continued to explain when he received the Nobel Prize in the 1930s, his Nobel lecture in 1934, that indeed in the beginning things were taken as being more simple than what they were, but at that time it was already clear that development and heredity were much more complicated than what was initially thought. And indeed, the 1930s was a time when many geneticists started thinking differently about both eugenics and heredity. But this was not the case in Nazi Germany, where Mendelian genetics were adopted to explain the inheritance of several traits that were not considered as favorable, one of them being blindness. And there were pamphlets and leaflets to explain how several social traits could be avoided by achieving pure homozygosity, always citing, as you can see here, typical Mendelian genetics. Even Godard's book, The Calical Family, was retranslated and republished in 1933 as soon as the Nazis took power. And from then on, many people that were involved in legislation, like Arthur Gutt, you can see here, were determined to show that there are ways in which specific traits are transmitted according to Mendelian rules. That's himself saying that. And he was the one to develop a memo for what came to be known as Nuremberg laws, in which he began again with a typical example of Guinea pigs and inheritance. And he suggested, and this is what the laws ended up saying, that being Jewish was a recessive Mendelian trait, and this is what should be taken into account in which kind of marriages would be allowed. Bernhard Lochner, who was a Nazi as well, nevertheless reacted saying that, look, we cannot do what people in the United States did, apply what was known as the one-drop rule. We cannot leave out all people with some Jewish ancestors, because in this way we will alienate their German relatives, and look at the last point, we lose approximately 2 divisions of soldiers. So they arrived at the compromise. Their model was the US laws, but they found some of them very harsh. And they arrived at the Nuremberg laws, which you can see here, they are represented in a typical Mendelian way, in which they made a distinction between the pure Aryans, the pure Jews, and people in between who might have 1 or 2 Jewish ancestors. And these people were accepted to stay in the community insofar as they were not married to other Jews and they did not practice the Jewish religion. So we see here that the main concern for the Nazis was to prevent the unwanted results of what they called Herausmendel or Mendeling out, the sudden reappearance of what we know as recessive traits. And this is what they promoted initially, purity via homozygosity. Of course, as you know, when the war started, things took a very nasty, very different direction. But the initial plan was to clear society from what they perceived and they justified as a typical Mendelian recessive trait. And of course, all the propaganda that they used against not only Jews, but also all kinds of people, including feeble-minded people, was based on their understanding of US laws, which they cited and followed very, very, very carefully. And this is how they decided to implement their own laws, noting that in America, black or colored is not only used to describe a full-blooded Negro, but also anyone with even a drop of Negro blood in their veins, who is thus excluded from the white community. This is the one-drop rule in the US South. They found it very harsh, and they decided not to use that. Of course, they did worse things later. And this made people like the Jornet furious. And in 1934, he commented that the Germans are beating us at our own game and are more progressive than we are. Because whereas a certain number of sterilizations took place in the United States, 60 to 70,000, that's a lot, but a lot more had taken place in Nazi Germany only in a few years. Several other people in the United States became proponents of Hitler's and his policies. This is Whitney that we saw earlier with the Mendel Theater. This was another Norwegian eugenicist who tried to show that we should stop the mixing of races because they increase mortality from tuberculosis, which is an infectious disease, but it didn't matter to him. And how did they manage to do that? And this is the part that is of interest to us. They integrated the teaching of Mendelian genetics in schools along with racial science. And here you see the drawing by a young girl that explains in typical Mendelian genetics terms how we can figure out whether a person is a Jew. And this reminds us, of course, of similar approaches today, but that's a topic of another book. I've written how we identify nationalities and ethnicities. I should just say they're not in our DNA. It is a more complicated thing. But the point is that in Nazi Germany, racial science was taught along Mendelian genetics, as you can see here and here, with the purpose to promote their ideology and to make discrimination among different kinds of people. And they use typical Mendelian genetics, classical illustrations to make their point about who is a mischling, a mixed person, and who is a pure one. And they refer to feeble-mindedness and to how dangerous it is for this kind of trait to be transmitted across generations. There is actually a study published in PNAs that shows that many people who were educated, schooled in the 20s and the 30s had more severe attitudes against Jewish people. Here you can see a gap because many of them died during the war. They were soldiers. But the most important conclusion was that they remembered as their main influence not the Nazi youth clubs, but schools. And now if you're wondering why and how people in Germany followed the Nazis, the answer is simple. Biology and the teaching of biology and racial science in schools seems to have been used as a means for indoctrination. And this is the dangerous aspect. So my question is, with all this history, why do we teach this in schools instead of something more realistic, more authentic? And my suggestion is that we should be teaching about development. And so, whereas if you ask students in schools why these two plants differ, and they will tell you that the one on the right it has a dominant allele and the one on the left has the recessive allele, the real answer is that for these plants of Arabidopsis thaliana, these are clones, but they have been exposed to different environmental conditions. People who are touching the one on the left like animals do when they browse on plants, and this resulted in a different development. That's a typical case of phenotypic developmental plasticity. And so my suggestion is that instead of teaching that genes determine phenotypes, we should introduce the black box of development, which is exactly quite black. There's a lot that we do not know, but nevertheless, I believe that we can give students a sense of complexity. How we can do this? I can propose, and this is what I do in the book, at least 4 metaphors that we can use in classrooms. So the first one is that if we're talking about DNA, which usually students perceive as a recipe for doing something, we can imagine of two developmental systems that can result in two different phenotypes, and I have empirical evidence for both of these. The point, therefore, here is that whatever is in DNA alone does nothing, but how the information encoded therein will be used depends on the developmental system that will use it, with a lot of variation possible. Another metaphor has been proposed by Louis Wolpert, and it is the origami metaphor, according to which what we have in genes are origamis that do not tell us how the adult form will look, but how we can produce the adult form. And this is very important because it takes into account whatever can happen during development. A third metaphor was proposed by Evelyn Fox Keller, who argued that if environment and genes made independent contributions in the way that these two children pour water into the bucket independently, we might be able to separate their contributions. But at the individual level, we cannot, because they're both necessary. One is opening the tap, the other is pouring the water, and there's no way at the individual level that we can separate anything. And this is why there can be variation during development. Finally, the last metaphor, when we move to the population level, a mistake we make is that we identify a difference maker and think of them as causes. So if there is snow on the North Pole, but not in the South Pole, we can think that high humidity in the first was the cause of the snowfall, whereas low humidity did not allow for snowfall in the South Pole, but we tend to forget that low temperature is also necessary for snowfall. It is just that we have low temperature at both places, that this does not make any difference, but this does not mean that this is not a cause. I analyze this further in the book. My argument is that we should be teaching heredity, the phenomenon itself, not genetics, the science that studies the phenomenon. And with this, I thank you very much for your attention, and I'm happy to answer any questions that you may have, which I believe you can write in the chat.